Okay. Can one of you unmute and let me know if my slide is visible and if I'm audible? Okay, thanks for typing in the chat. So, good morning. Today is the 9th live session of the series of live sessions conducted for this course, Fundamentals of Protein Chemistry, via course by course instructor Professor Shagota from IIT Kharagpur. So in these live sessions, what we do is that we basically discuss some of the previous year assignment questions, and we try to figure out not only the answers of the questions but also the concepts behind those questions, so that it helps us solve similar type of other questions, right? So and how we want to conduct this session is that we try to make the sessions as interactive as possible, so that if you are not able to understand any answer or if you have any query, you can just unmute and let me know and we can discuss, or you can type in the chat box and I will answer you. And if I know the answer right now, I'll tell you right now. If I don't know at the moment, I will definitely learn about it, read about it, and let you know by next class. Okay, that is how we basically conduct these live sessions. Alright, so we'll start today's session with the first question for today. Okay. My name is Aishi and I am a PMRF research scholar from the Department of Chemistry, IIT Bombay. Okay, and today is the 9th live session. So we'll move on to the first question from the set of questions that we have for today. So this is the first question. The protein stability curve delta G versus temperature typically appears as, so there are 4 options. If you see, there are 4 visual representations of graphs, right? A, B, C and D, right? So you have to tell me which is the correct representation of protein stability curve delta G versus temperature, right? These are the 4 options. I will give you a few minutes. I want you to think about it and come up with the answer. If you can, just unmute your mic and let me know. You have to give me the detailed explanation as well, just not the answer. Okay? And then we can discuss. I'll give you a few minutes to tell me the answer. Anybody? Anybody with the answer? Anybody just try to answer? Okay, so the right answer will be A. Now we'll discuss why will it be A, right? So knowing an answer is not enough. We need to know why that answer is the correct one, right? So here we are talking about the protein stability curve or delta G versus T, right? And how is it typically shown? So in that case, we need to know a little bit about um delta G of the unfolding. So y-axis will be free energy of unfolding delta G, right? And x-axis will be temperature in degrees Celsius or Kelvin. Now the typical feature of the curve will be an inverted U shape because it will be a parabolic shape because proteins are more stable at an intermediate temperature, which is delta G maximum, right? And at both low and high temperatures of temperature, the protein becomes less stable, that is delta G decreases. So delta G is 0 points where the curve crosses the x-axis, delta G is 0, right? Protein is half unfolded. These are the cold denaturation temperature and the heat denaturation or melting temperature, right? And peak stability, the top of the curve shows the temperature at which the protein has maximum stability, delta G max. So protein stability curve is typically an inverted parabola with delta G on the y-axis and T on the x-axis, showing cold denaturation on the left, and heat denaturation on the right, and a maximum stability point in between. Sorry, the laser pointer is malfunctioning. Yeah, so there's some. Yeah, no, it's recording. Yeah. Just give me a minute. There's some issue with the Google Meet. Okay. Okay. Uh, yeah, so not a problem. Yeah. So, yeah. So, as I was saying, sorry for the disruption. As I was saying, so protein stability curve is basically a inverted parabola, inverted U shape, with delta G on the y-axis and temperature on the x-axis, showing cold denaturation on the left, heat denaturation on the right, and a maximum stability point in between, somewhere here. Okay. So, we'll move on to the next slide. So this is the NPTEL course with Dr. Pramit Chaudhary, and the slides here are very relevant to what we have been studying. So I would like to explain from here as only well. So here he has talked about protein stability curves. So the stability curve of a protein is defined as a plot of the free energy of unfolding as a function of temperature. That is explained, right? So that they define stability curve as essentially that the dependence of free energy on temperature. And whatever we are looking at, whatever is free energy of denaturation, where T stands for denatured, N for native. We're looking at the difference in free energies between the denatured and the native states. So you see, its stability curve for protein, one of the major points in this case was, if you would compare it with the stability curve or with the free energy curve of a hydrophobic molecule being dissolved in water, then you will see the difference, being that TH and TS are very near to each other. Okay, what are TH and TS? If you can see from here, okay, TH and TS, right, are very near to each other, will be very near to each other. And in this case for a protein, however, for a hydrophobic molecule like neopentane, this TH and TS are far apart from each other, right? So there are a couple of more points in this. Sorry, there are a couple more points in this case, right? Um, if you look at the stability curve, which is this plot of again delta G versus temperature, there are 2 points at which the stability curve cut the zero line here. So this is the zero line. So there are 2 points. So when the delta G is equal to 0, it is essentially, that means K is equal to 1. K equilibrium is equal to 1. What it means that we have equal concentration or equal number of unfolded molecules and folded molecules, right? And that is referred to as a midpoint of any transition. What I refer to, what I mean by midpoint will be more clear in just a few minutes. But the more important thing is that we see 2 points. One is we know that when you have a protein heat up, it denatures. That means you destroy all the stabilizing interactions and you start opening up. So one of those points is Tg, which is the heat induced. That means increasing the temperature. But what possibly we did not realize was that if you would also decrease the temperature, a point would come where the protein will again denature, right? So that means a point will come where k will be equal to 1 again. So that is your Tg prime, which is the cold denaturation point. Okay. So, if you recall the thermodynamic equations, we had looked at the thermodynamic relations before in one of the sessions, and these are some of the relations we are going to be using, quite often in this field. Now, properties of a stability curve. So the native protein is assumed to be stable in a certain range of temperature, right? Hence delta G for unfolding will be positive in that temperature range. The stability curve crosses the zero line at the points. Now these points are the low temperature and the high temperature melting points and are noted by Tg prime and Tg respectively. Okay. So why these two are very close to each other for a protein? So delta Cp for unfolding is large, okay, and falls in the range of 12 to 18 degree per residue. So maximum stability of proteins tends to be small, on the order of 50 to 100 calories per residue. Hence Ts and Th are only within a few degrees. Okay, so we don't have to get into more thermodynamics of this. Basically, you have to remember that protein gets denatured at both lower temperature and higher temperature. Okay, we won't get into too much of these course. If you want, you can just go and check out the actual lecture by Professor Pramit. So, what we started with was the question, which one is the proper protein stability curve and how does it appear. So, the plot is typically an inverted parabola, as I said, inverted U-shaped. As you can also see from the right answer is this one. And this shape arises because the free energy change depends on enthalpy and entropy, both of which vary with temperature, right? And through the heat capacity change, that is the delta Cp that we were talking about, between folded and unfolded states, right? So what would be the key features? Delta G max occurs at an intermediate temperature, T optimum. Protein is the most stable here, right? Cold denaturation is this Tc. Okay. On the left, at low T, delta G drops to 0. Below this point, proteins unfold because hydration of non-polar groups is favored over hydrophobic collapse. Right? When I was explaining, I probably made a mistake and said this is the cold denaturation and it's a hot. That's wrong. This is the cold denaturation. This is lower temperature, so cold denaturation, and this is higher temperature, so heat denaturation, right? In cold denaturation, below this point, proteins unfold because hydration of non-polar groups is favored over hydrophobic collapse, right? And then heat denaturation will be this on the right. Delta G again drops to zero. Above this point, thermal energy dissipates, disrupts non-covalent stabilizing forces. And what will be delta G 0 point? So Tc, Tm, folded and unfolded states are equally balanced, populated, like 50% unfolded. A typical annotation of a published curve, labels on the curve will be Tc, cold denaturation temperature here, and Tm, right intersection with the x-axis, melting temperature. Delta G max will be peak stability. So arrows often indicate stability window where protein remains folded. Right? Sometimes the folded versus unfolded population fractions are drawn as separate sigmoidal curves beneath the delta G plot. Okay. So that is why A will be the correct answer. Okay. I'll wait for a few minutes. If you have any questions or any queries regarding this, you can ask me right now. Then we move on to the next question for today. Anybody, any questions? Guys? Okay, so if there are no queries, we'll move on to the next question for today. Which of the following statements about cis and trans peptide bonds in proteins is correct? Okay, I'll repeat the question for you. Which of the following statements about cis and trans peptide bonds in proteins is correct? Option A. Cis peptide bonds are more common than trans peptide bonds in proteins. Option B. The omega angle in trans peptide bond is approximately 0 degrees. Option C. Cis peptide bonds are frequently found preceding proline residues. Option D. Trans peptide bonds are energetically unfavorable and rarely occur in proteins. I'll give you a few minutes. You can think about it and come up with the answer. And you unmute and let me know with the proper explanation, detailed explanation, not only the answer. And then we'll move on to the next question for today. Anybody? I'll give you 2-3 more minutes, then we'll start discussing. Okay, so we'll discuss the answer now. Let's talk about the first sentence. Cis peptide bonds are more common than transpeptide bonds in proteins. False, because trans is much more common, over 99%. Like cis is very rare. Next statement, B. The omega angle in transpeptide bond is approximately 0. Also false, right? Because in the trans conformation, the omega angle is approximately 180 degrees Celsius. In the cis, omega is 0 degrees Celsius. Sorry, 0 degrees, not Celsius. Let's talk about option D. Transpeptide bonds are energetically unfavorable and rarely occur in proteins. Again, false, because trans is energetically favorable and dominates in proteins. So the correct answer is C. Cis peptide bonds are frequently found preceding proline residues. Okay. Now, what is a peptide bond? A peptide bond is a covalent bond between the carbonyl group C double bond O of one amino acid and the amide nitrogen NH of the next. Now, because of the resonance, the CN bond has partial double bond character, like planar, restricted rotation. So, the omega dihedral angle defines the torsion around the CN bond. Cis versus trans geometry. Trans peptide bond will have omega 180 degree. The alpha carbons of adjacent amino acids are on opposite sides of the peptide bond. So this minimizes steric clashes and is more energetically favorable. Over 99% of peptide bonds are trans in proteins. However, for the cis peptide bonds, the omega is approximately 0 degrees. The alpha carbons of adjacent residues lie on the same side, causes steric hindrance between side chains, energetically much less favorable and found in less than 0.1% of non-proline peptide bonds. In special cases like proline has a cyclic side chain that bonds back to its own backbone nitrogen. This rigid structure reduces the energy differences between cis and trans conformations. So as a result, about 5 to 10% of XPRO bonds when the next residue is proline are cis. So this is 100x more frequent than cis bonds before other amino acids. So cis-pro bonds often play important roles in protein folding, turns, and regulations, and are acted upon by enzymes called peptidyl prolyl isomerases. Right? So we have discussed about the structure of proteins a lot. So I just will go through it very fast. 20 amino acids are there commonly found in proteins and are joined together by peptide bonds. The linear sequence of the linked amino acids contains the information necessary to generate a protein molecule with a unique three-dimensional shape, and the complexity of protein structure in 4 organizational levels: primary, secondary, tertiary, and quaternary. Now, these are the 4 levels of protein organization. Primary is in sequence of a chain of amino acids. Secondary occurs when the sequence of amino acids are linked by hydrogen bonds. Tertiary occurs when certain attractions are present between alpha helices and beta sheets. And Quaternary is a protein consisting of more than uh, one amino acid chain, right? So the primary structure is defined as a sequence of amino acids, nothing. It's just a straight, it's not straight, just simple, planar, basically the sequence of amino acids, nothing, no structural configurations involved. So understanding the primary structure is important. Why? If it's a simple, most simple level of protein, then why is it so important? Because many genetic diseases arise because of mutations with mutations in the gene and abnormalities in this primary amino acid structure, right? Which causes error in folding, loss of impairment of normal function, all of that. Now, peptide bonds, again, we have discussed in proteins, amino acids are covalently joined by peptide bonds. So, these bonds are amide linkages between the alpha carbonyl of group of one amino acid and the alpha amino group of the another. And for example, the formation of peptide bond between glycine and alanine. Peptide bonds are broken by prolonged exposure to strong acid and base at high temperature to hydrolysis, and these bonds non-enzymatically. So this is the peptide aramide synthesis. So naming free amino acid in the N terminal written to the left and free carboxyl is written to the right. Therefore, all amino sequences are read from the N terminal to C terminal of the peptide. And polypeptide is basically linkage of many amino acids through peptide bond results in an unbranched chain. So residue or moiety, each component amino acid in a polypeptide. So when a polypeptide is named, all residues have their changed to YL with the exception of C-terminal amino acids. Now, what are the characteristics of the peptide bond? Partial double bond character, that is, it's shorter than single bond, rigid and planar. This prevents the free rotation around the bond between the carbonyl and the nitrogen of peptide bonds. Trans configuration because of steric interference of the R groups when in the cis positions and uncharged but polar. Now, determination of the amino acids composition of polypeptide. The determining of the primary structure of polypeptide is to identify and quantitate its constituent amino acids. Okay, by hydrolysis with a strong acid at high temperature for 24 hours to release the individual amino acids which can be separated by cation exchange chromatography, right? So mixture of amino acids bind to the column that contain a resin which negatively charged group with different with different affinities depending on their charge and hydrophobicity. The separated amino acids contained in the eluate with solutions of increasing ionic strength are quantitated by heating them with anhydrine, a reagent that forms a purple compound with most amino acids. The amount of amino acids is determined spectrophotometrically by measuring the amount of light absorbed by the anhydrine derivatives. So this analysis is described as performed using an amino acid analyzer, right? We'll not go into sequencing. We have already discussed about this in one of the previous sessions. Now the secondary structure is defined as regular arrangements of amino acids that are located near to each other in the linear sequence. Like alpha helix, which is a spiral structure consisting of tightly packed coiled polypeptide backbone core with the side chains of component amino acids extending outward from the central axis to avoid interfering serically with each other. The telephone cord shape of the alpha helix is held in place by hydrogen bonding between every NH group and the oxygen of a carbonyl group in the next turn of the helix. 4 amino acids down the chain. So I and I plus 4. The typical alpha helix is about 11 amino acids long. So a very diverse group of proteins contain alpha helix, like the keratins, fibrous proteins, major component of tissues such as hair and skin. Alpha keratin and myoglobin is globular flexible molecule whose structure is approximately 80% alpha helical, and their rigidity is determined by the number of disulfide bonds between the constituent polypeptide chains. Now, hydrogen bonds alpha helix is stabilized by extensive hydrogen bonding between the peptide bond carbonyl oxygens and amide hydrogens that are part of the polypeptide backbone. The first and the last peptide bond component are linked to each other through hydrogen bonds. Hydrogen bonds are weak, but they collectively serve to stabilize the helix. So each turn of an alpha helix contains 3.6 amino acids. The amino acid residues spaced 3 or 4 apart in the primary sequence are close together and folded in the alpha helix. Now, amino acids can disrupt an alpha helix. Proline disrupts an alpha helix because its amino group is not geometrically compatible with the right-handed spiral of the alpha helix. Charged amino acids disrupt the helix like glutamate, aspartate, histidine, lysine, or arginine. By forming ionic bonds or by electrostatically repelling each other, amino acids with bulky side chain like tryptophan and a large number of amino acids, example valine or isoleucine that branch at the beta carbon, can interfere with the formation of the alpha helix. Now, beta sheet is the second form of regular secondary structure in proteins. They are less common than the alpha helix. Structure of beta sheet appear pleated. Okay, so comparison of beta sheets are more composed of 2 or more peptide chains. Hydrogen bonds in beta sheet are perpendicular to the polypeptide backbone, and parallel or antiparallel sheets can be formed from 2 or more separate polypeptide chains that are arranged either antiparallel or parallel. Polypeptide chains are arranged to each other with the N-terminal and C-terminal ends of the beta strands. Polypeptide chains are arranged to each other with the other N-terminal. So you see the difference between the parallel and the anti-parallel beta pleated sheet. Now there are interchain bonds as well. The interchain bonds, the hydrogen bonds are formed between the polypeptide of the separate polypeptide chain, between the polypeptide backbones of the separate polypeptide chains. So interchain bonds, a beta sheet can also be formed by single polypeptide chain folding back on itself. In this case, the hydrogen bonds are interchain bonds. So, parallel, anti-parallel. So, what are reverse turns or beta bends? So, bends are reverse the direction of a polypeptide chain, helping it form a compact globular shape, right? So, they are usually found on the surface of protein molecules and often include charged residues. Beta bends generally composed of 4 amino acid that causes a kink, and the amino acids with the smallest R group. So, beta bends were given this name because they often connect successive strands of antiparallel beta ends. So, they are stabilized by the formation of hydrogen and ionic bonds. What are non-repetitive secondary structures like ends, loops, and turns? So they are best recognizable as forms that resemble neither an alpha helix nor a beta pleated sheet. These do not have repeating element in their amino acid sequence. So these structures are often found on the surface of the proteins. Now, what do you mean by super secondary structures or motifs? So, you must have used or heard or used the word motif a lot while studying biology. So, structural biology specifically. So, super secondary structures are usually produced by packing side chains from adjacent secondary structural elements close to each other like alpha helix and beta sheets, they are adjacent in the amino acid sequence are also easily adjacent in the final folded protein. Okay, so some common structural motifs combining alpha helix and beta sheets are these like beta meander, beta barrel, Greek key, and all of that. So, motif is an arrangement of a small number of secondary structures recurring in many different proteins. A motif is a short recurrent segment of a protein's three-dimensional structure, very important, that is found in a large number of different proteins. The motif may contain 2 to perhaps 7 or 8 elements of secondary structures. Like the example is a beta alpha beta motif consists of 3 secondary structures, a beta strand connected to alpha helix connected to another beta strand. So motifs cannot exist on their own, they require the rest of the domain to maintain their conformation. Tertiary structure, very important. Tertiary structure is the total 3D conformation of the entire polypeptide chain, including alpha helices, beta sheets, and loops or turns or whatever bends. The structure of the globular proteins that we have talked about in aqueous solution is if we talk about aqueous solution is compact with high density of atoms, right? Atoms in the core of the molecule. Hydrophobic side chains are buried in the interior, whereas hydrophilic side chains are seen on the surface of the protein, the molecule. This eliminates the possibility that water molecule may become bound to the hydrophilic groups and disrupt the integrity of the protein. Now the alpha helix and the beta sheet provide maximal hydrogen bonding for peptide bond components. Peptide bond components within the interior of the polypeptide are involved in hydrogen bonds. So what is the structural domain? Structural domains are the fundamental, functional and three-dimensional structural units of polypeptide. The polypeptide chain contains two or more domains, and the core of the domain is built from combination of the motifs. Now, disulfide bonds, what are the interaction stabilizing tertiary structure? So, the disulfide bond is a covalent linkage formed from the sulfhydryl group of each of the two cysteine residues to produce a cysteine residue, right? So, disulfide contributes to the stability of the protein tertiary structure and prevent them from denaturation in the extracellular environment. Amino acids with non-polar side chains tend to be located at the interior of the polypeptide molecule where they are associated with other hydrophobic amino acids. In contrast, amino acids with polar tend to be located on the surface in contact with the polar solvents. Now, amino acid side chains containing oxygen or nitrogen bound hydrogen can form hydrogen bonds with electron rich atoms such as oxygen of a carbonyl or carboxyl group, right? Of a peptide bond. Formation of hydrogen bonds enhances the solubility of the protein. Now, ionic interaction like negatively charged groups such as the carboxyl group in the side chain can interact with positively charged groups such as the amino group in the side chain of a polypeptide. Protein folding also we have discussed in one of our previous classes, which occurs within the cell in seconds to minutes. Implies a shortcut through the maze of all folding possibilities. As a peptide folds, its amino acid side chains are attracted and repulsed according to the chemical properties. We will not go into the details of chaperones because quaternary structure of protein is the arrangement of multiple folded protein or coiling protein complex in a multi-subunit complex. We have discussed about denaturation. We will not go into these things because we have already discussed it. One second. Okay. So nothing to discuss in this aspect. We know that amino acids are chiral. We have discussed about it. Which amino acid is not chiral? If one of you can answer. Glycine. Glycine is not. So if you see, the chemical properties of the peptide bond are important for determining the shape of the polypeptide chain. Some of the bonds of the peptide chain are free to move, rotate, but the peptide bond itself can only adopt certain conformations. To understand why, we return to the topic of the orbitals that surround atoms. When a covalent bond forms, the orbitals of the two bonded atoms overlap and mix, creating a bonding orbital that holds the shared pair of electrons. Bonding orbitals are combinations of atomic orbitals from each atom, and much like atomic orbitals, they describe where the shared electrons are likely to be found. A single bond, sometimes called alpha sigma bond, is composed of a cylindrically symmetrical orbital that joins two nuclei. Double bond consists of one sigma bond as well as a second bond called the pi bond. The pi bond forms the p orbital of two nuclei overlap. This is plus 2 knowledge. Yeah. So peptide bonds always favor the trans configuration, you see, because the peptide bond is not able to rotate freely, right? So it can exist in two possible conformations known as the geometric isomers. To understand the concept of geometric isomers, let us first consider the simple case of the hypothetical molecule shown in figure 16a. Okay, 16a will be okay. Anyway, this is from a book. It can exist as two geometric isomers such as cis and trans. Now, which describe how two atoms or groups of atoms on opposite sides of the double bond are oriented relative to one another. Two atoms or groups of atoms are cis to each other where they are on the same side of an imaginary line drawn along the length of the double bond. If they are on opposite sides of the imaginary line, they are trans to each other. Likewise, peptide bonds can exist as cis or trans isomers depending on whether the alpha carbons of adjacent amino acids are on the same side of the peptide bond or on the opposite sides across the peptide bond connecting them. In most cases, the trans isomer is favored over the cis isomer. The reason for this is that the cis isomer places the R groups of two connected amino acids in close proximity to each other. This results in a steric clash, a non-bonded interaction in which the electrons involved in a bond get too close to electrons involved in an adjacent bond. This concept can be understood intuitively as electron clouds of two chemical groups trying to occupy the same space at the same time and bumping into one another. As you might imagine, steric clashes are not favorable. In the trans isomer of the peptide bond, the steric clash is avoided and the R groups are held far too apart. The extent to which trans configuration is favored over the cis configuration is dependent on the chemical identity of the R groups, as we will see. We will not go into too much. So if you see, Yes, so side chains in a polypeptide chain determine how it folds into three-dimensional structure. This occurs in part because the amino acid chains differ in their size and flexibility, which in turn biases the peptide backbone towards certain conformations. Some amino acids have large, bulky side chains like phenylalanine and tryptophan, whereas others take up too much less space like alanine and glycine. The side chains of some amino acids are also more flexible than others, like methionine, for example, can adopt many more conformations than isoleucine or valine, because of which a methyl group on the side chain close to the peptide backbone on the beta carbon, the second carbon from the carbonyl carbon, this methyl group restricts the conformations available to the peptide backbone because it creates steady clashes where certain conformations are adopted. So, glycine and proline have an even more pronounced effect on the peptide backbone. Glycine effectively lacks a side chain. As such, it enables the peptide backbone to adopt a broader set of conformations along the two non-peptidic bonds and the repeating unit of the peptide backbone. Proline side chain, which is connected to its amino group, prevents rotation around the bond connecting the amine and the alpha carbon. So, most amino acids from form peptide bonds that strongly favor the trans configuration because of steady clashes in the cis configuration. Proline is an exception because its side chain causes steady clashes in both the trans and cis configuration. As a consequence, peptide bonds in which the nitrogen of the peptide bond comes from proline sometimes exist in the cis conformation. Okay, we'll not go into too much detail. We already discussed this and these we have already talked about. I'll upload the slides. If you want to go into details, you can just go through these slides. We have discussed Ramachandran also in two classes, so I'm not going to go into details of Ramachandran plot as well. Coming back to the question that we started with, the correct answer will be cis peptide bonds are frequently found preceding proline residues. Do you have any question? If you have any question, you can just ask me right now. If you don't, then we'll stop for a few minutes and then we'll move on to the next question for today. Okay. I'll wait for a few minutes for you to ask doubts. you don't have any questions, we can move on to the next question for today. You have to link each of the images to the secondary structure that is. So these are the options: beta hairpin, coil, zinc finger, and Greek key. Which is which? I'll wait for a few minutes for you to think and come up with the answer. Then we'll discuss this question. So we'll discuss this question. So the answer is the first one is Greek key, second one is coil, third one is zinc finger, and D is beta hairpin. Now we know about the chemical structure of the amino acid, nothing new. So we know about the different amino acids, all of that. There's nothing new over here also. We know about the biophysical characteristics also. We have positive charge, negative charge, neutral, polar charged, polar uncharged, aromatic, and all of that. So we have already talked about the unity, diversity of proteins, the four levels of. We have talked about denaturation, all of this. So if you see alpha helix, no, not this one, sorry. Alpha helices can wrap around one another by interactions between the hydrophobic side chains to form a stable coil structure. Okay, so this is a coil structure. Okay, so you already see which one is the coil. Okay. Beta sheet alpha helix, nothing new over here. Gel electrophoresis is used to separate proteins. Know about all of this, it's just a repetition. Nothing computational we are going into. Okay, so these are the proteins and their functions. Nothing related to the questions. We'll go to the slide that is. Okay, so if you see over here, alpha, beta, this is called Tim barrel, this is a sandwich, this is roll. These are all examples of different types of combinations of secondary structures, right? Alpha beta alternating, parallel beta alpha beta units, anti-parallel beta sheet segregate, all of these alpha plus beta. Different types of classes, different folds, different architectures. This is called the Tim barrel fold. You know about PDB. PDB is very important. Any structure that has already been determined using X-ray crystallography or cryo-EM or whatever, NMR, they are already uploaded on this website. Basically, you publish papers and you also deposit a particular ID, deposit the structure to this database, which gives it a particular ID. discuss this. Okay, sorry. This is the Greek key motif. Okay, if you see, this is what we call an N-terminal Greek key motif. Kind of, you see, an anti-parallel, all of these. So the Greek key motif is a beta sheet topology involving 4 beta strands connected by loops in a characteristic order, right? Topologically, the connectivity between the strands is often described in terms of +3 -1, as the fourth strand is adjacent to the first via a longer loop that we can see over here, right? Fourth strand is this one, adjacent to the first strand via longer loop. Longer loop is this. In effect, 3 of the strands are connected via hairpin loops and the fourth is linked in a crossover fashion to the first and third strand by a longer loop. The Greek key motif is very common in beta barrels and beta sandwich structures. In the image panels, if you see, you'll see one representation of the Greek key. So, examples will be vitelline, membrane, outer layer, protein, and all of this. Greek key, more generally, many beta barrel proteins and beta sandwich proteins uses the Greek key motif. Another classical example is superoxide dismutase, which has beta strands arranged in a Greek key topology in its beta barrel features. Beta barrel core. So this is the alpha helix motifs. If you see alpha turn helix, helix loop helix, and this is the coil. We already discussed about coil in one of the previous slides. So coil is formed when 2 or more alpha helices wind around each other, forming a superhelical structure. So the helices usually have a repeating heptad pattern where residues at positions E and D tend to be hydrophobic, and these hydrophobic residues from different helices interlock like knobs into holes packing, which stabilizes the interface. And coils are common in structural proteins, transcription factors, and oligomerization domains. Okay, so examples will be many synthetic coils are used in design as molecular interacting peptides. In nature, myosin has long coil tails. Transcription factors like leucine zippers are also coils. Okay, then we talk about beta hairpin. It's one of the simplest beta structure motifs. 2 antiparallel beta strands connected by a short loop or turn. The loop may often be turned favoring residues of glycine or proline to facilitate the sharp reversal in direction. The 2 strands form hydrogen bonds with each other. So minimal example is tryptophan zipper. Okay. Then we have the zinc finger, which is a small structural motif in which a zinc ion coordinates with side chains like commonly cysteine and histidine to stabilize the fold, and often comprises a beta hairpin and a beta alpha helix. The canonical C2H2 zinc finger motif, 2 cysteine residues bind zinc, then a beta hairpin region, then an alpha helix, then 2 histidine residues also bind zinc. So total coordination will be cis2 his2, and the helix often lies in the DNA major groove when the finger binds DNA, right? So the zinc ion acts as a structural cofactor, stabilizing the small fold and allowing the motif to present a rigid helix for DNA interaction. So this is Greek key, this is coil. Okay. This is the zinc finger and this is beta here. Okay. Anybody has any questions regarding this? You can ask me now. I'll give you a few minutes. Okay, so if you have no question, maybe we can stop the meeting here for today and we'll meet again this Monday, that is tomorrow. Thank you, everyone. Thank you.